Hi, this is Kelly Busby. This is the second part of this lecture. Phase 1, we have different phases of research. Phase 1 clinical trials, the biological question is, it is safe. This is when the safety is tested generally on small numbers of healthy volunteers after animal studies have established that the drug is not completely dangerous. After the side effects are recorded and the drug therapy is deemed worth the risk by balancing the risk of the disease with the risk of the drug, they may move to studying sick persons to establish the best dose of the drug. Balancing the risk of the drug against the risk of the disease is subjective. While it's completely unacceptable to have hair loss during administration of a drug for simple headaches, obviously hair loss on an acceptable risk for a cancer patient who faces death or disfigurement is an acceptable risk. The most likely route of administration are determined in these first phases, as is the dose that seems to do what we want the drug to do without the worst side effects. Phase 2, does it work? At this level, more people are studied, and we're looking at efficacy. There generally can be up to 100 persons at this level of research who get the dose that was selected after the phase 1 studies determine a safe and hopefully effective dose. Effects of the disease process are the dependent variables that we recorded. At this point, there needs to be a number of persons who get a placebo to compare with the ones who got the dose. They are, of course, the control. Once the FDA has determined that enough persons benefit, the next phase is started. Phase 3, is it better than what we have now? At this level of research, all persons in the study will be getting a drug or a therapy except for the controls who would get placebos. There could be thousands of persons studied at this level, and the researchers are comparing the new drug to an existing drug on the market that has a known track record. The biological question is this new drug better than the existing drug? Needless to say, these comparisons may be done with more than one existing treatment. Just like Phase 1 and Phase 2 studies, Phase 3, drug participants are watched closely to see if any serious side effects develop. Treatment is stopped if side effects appear to be dangerous. If a drug treatment is shown to be just as effective but with less side effects or more effective than existing therapy, the FDA approves the drug for general use. At this point, the drug is given its trade name and is put on the open market. Now, Phase 4 trials, are there any other uses or benefits? Sometimes this is called the post-marketing phase, in that the researchers are starting to branch out. If it works on F and D is a similar disorder, will a drug therapy work on D also? In addition, the post-marketing stage is critical for drug interactions, because once a patient is buying the drug, there will be situations that arise. Once a patient is buying the disease, there will be situations that arise that didn't happen in the clinical trial. A famous example of the importance of keeping records during the post-marketing phase is the fen-phen episode, which occurred in the 1990s when two short-term appetite suppressants were studied separately, and they were both found to be safe and effective, and they went on the market pretty much at the same time. Once the two diet drugs went on the market, they started getting prescribed together. And unfortunately, this effective drug combination caused as many as 20% of the dieters to develop a primary pulmonary hypertension that caused them to become permanent pulmonary cripples and shorten the lives of many of these patients. Thank you.